Hello, everyone, and welcome to today's webinar. My name is Mingjuan Liu. I'm the Director of Marketing at uBriGene's, and I will be your moderator today. So there are still 2 minutes before 12. Before we start, let's do a quick sound check. Please confirm if you can hear us by using the Q&A box in the top right corner of the webinar platform. So there's a Q&A box. Okay, I see some thumb up. Okay, thank you. Thank you so much. Will we just 1 more minute to let more people join in? Thank you for your patience. Okay, hello again. Now it's on top of the hour. Welcome to the iPSC Innovation Webinar, Unlocking iPSC Innovations, Next Generation Reprogramming, Engineering, and Manufacturing Solutions. This webinar is a part of our series on advanced therapies, development, and manufacturing. We have a couple of webinar sessions previously on CAR-T, LVV, mRNA-LNP, and CRISPR. If you missed those sessions, you can watch the webinar recordings on our website. Before we dive in, I'd like to quickly go over a few logistics shown on the slide. So during the webinar, all attendees will be on mute, but we do encourage you to put in all your questions anytime by using the Q&A box in the top right corner. And then we will hold a Q&A session after presentation to address as many of your questions as possible. So now it's my pleasure to introduce the webinar speaker today, Dr. Scott Patterson. Scott is a PhD-trained molecular cellular biologist, as well as the seasoned business development leader. With over 17 years of experience spanning both research R&D and commercial strategy, Scott has played a key role in advancing therapeutic discovery and development across various modalities, including small molecules, biologics, cell and gene therapies, and genetic medicines. Before joining uBriGene's, Scott held leadership positions at a couple of biotech companies like QIAGEN, Discovery X, Fujifilm, Cellular Dynamics, Synthecal, and Maravai, where he contributed to strategic planning, alliance management, and corporate development. So today, Scott will share insights into next-generation iPSC innovations, covering cutting-edge advancements in reprogramming, engineering, and manufacturing. Hey, Scott, without further ado, I will hand over to you now. Thank you.
Thank you, Mingjuan.
You're welcome.
And thanks, everybody, for taking time out to learn more about our iPSC platforms. So I will hop right into the agenda slide. We've already done introductions. Just a quick introduction to uBriGene's Biosciences in case you're not real familiar with us, and then we'll dive into the slides around our iPSC solutions and leave room at the end for some Q&A. So uBriGene's is a global CDMO organization. They started about 10 years ago over in China, Lots of success in Asia. They have now, as you can see on the right-hand side of the slide, 4 GMP manufacturing sites. Cumulative square footage of GMP manufacturing space is around 250 to 300,000 square feet across those sites. And then we also have an innovation center in Nanjing. As you can imagine, as business took off, clients and prospective clients wanted to be able to do U.S. clinical trials. And so they pleaded with the leadership at uBriGene to please open a facility in the States so that it would make it easier for them to work with us to manufacture materials for their U.S. clinical trials. And so that led over here on the North American side of the map to the formation of our first North American company. So this is a Delaware incorporated U.S. company, started in Vancouver. That's still where our business headquarters are located. It's a pretty small team up there of HR, finance, regulatory affairs folks. And then we opened our first GMP site. It wasn't GMP when we opened it, but we opened our first site in the States in Germantown, Maryland, about 25 miles north of DC. Last April is when we signed the lease. We had that facility ready for GMP manufacturing by the end of July, and we started onboarding projects in September and currently have 2 or 3 projects in flight and 3 or 4 proposals under review. So gotten off to a pretty good start here in the States, considering we've only been open for business for 6 or 7 months. And one of the reasons that people are attracted to work for us is a couple reasons that are depicted on this slide. One is we'll meet you where your need is. So we're not just a GMP manufacturing CDMO, although that is our strength. If we work a lot with groups who are in startup mode, seed funded, Series A funded, they don't really have a process developed yet, and we'll do process development, analytical development around any of our GMP manufacturing platforms listed here. And that's another reason why groups like working with us is we have a pretty front to back solution for them, all the way from plasmid to viral vectors to cell therapies to genetic medicines, RNA, CRISPR assets, et cetera. And so once we've built credibility with groups and they're comfortable working with us on those PD, AD type projects, that streamlines very easily into GMP manufacturing of their assets, along with rigorous QC release testing and cold chain management and stability testing. The other thing that we won't talk about today, but that I've come to appreciate in my 8 or 9 months at uBriGene, is the team's ability to help clients get to first in human data very cost effectively and time effectively. And that's through the coordination of investigator initiated trials over in China. Suffice it to say that, our founding team has a great network over there of clinical CROs, GLP tox CROs, the tier 1 medical centers and the PIs working there, the regulatory agencies. And so we can help clients rapidly and very cost effectively move from promising animal model data, in vitro data, to first in human data. And that can get them to their next round of funding. And we'll probably have a webinar about that at some point, but we can talk more about that if you wish under an NDA. So these are really the hallmarks of what makes uBriGene attractive to our clients. We just have an unrelenting commitment to quality, collaboration, and innovation. And I'm not going to read all of these bullets. We're going to jump into our iPSC platforms. But you guys will have access to these slides. But, in talking internally as well as externally to clients and prospective clients, these are the reasons that they find value in working with us. So these are the platforms. Today, we're going to focus our attention, obviously, on iPSC, iPSC generation, iPSC engineering, as well as iPSC cell banking. Also a big part of a lot of the allogeneic programs we support and iPSC-based programs we support leverage gene editing. And so we'll also introduce, just show a couple teaser slides about our CRISPR-Cas capabilities. I presented a full webinar on that back in December, and you can find that archived webinar on our website if you like. And likewise, you'll find that we'll talk a significant amount about our reprogramming cocktail. That's an RNA-LNP formulated product. And we also did my former colleague, Ken Warrington, did a webinar on that back in September that you can get off our website. But we'll have a couple slides just to let you know about that platform if you weren't familiar with it previously. All right, so uBriGene's iPSC solutions. Typical workflow you'll see from the lower left-hand corner of this slide arcing up to the upper right-hand corner of the slide, from procuring the right cells from patients, to reprogramming, to gene editing, to actual clone screening, isolation and screening, and then banking. So we'll kind of use this slide as a roadmap for the webinar today. But before we jump into that, let's talk about some of the obvious challenges and bottlenecks to the clinical translation of iPSCs. And so that's this slide. The four that I've highlighted here are donor cell access hurdles. So it's a really, if you've never done it before, it's a very complex process with a lot of logistics. And we know that, and we've done that hard work for you and set up FDA compliant sourcing and screening infrastructure in order to make sure that we get the right high quality materials in-house to be able to execute on your project. And we'll have a slide or two on that. Another piece from a technical perspective is genomic instability. So a lot of the historic approaches for reprogramming fibroblasts or PBMCs into iPSCs leveraged viral transduction or transfection approaches that frequently led to integration and genomic instability. Our product, our mRNA-LNP reprogramming cocktail product, is what we use both for our internal processes, and we also offer it as a product if you want to do your own reprogramming. And it is an mRNA-LNP based formulation that is footprint free. It's non-integrating, and so you don't have that genomic instability concern with our mRNA-LNP reprogramming cocktail. The other big bottleneck that people worry about and that people spend a lot of hours on is screening and characterizing clones. Our mRNA-LNP programming cocktail and the protocols that accompany it are incredibly efficient. And so you'll see on a couple of the slides coming up, we typically get 4 to 10% of the clones that we screen after using our mRNA-LNP reprogramming cocktail are iPSC colonies, that pass our QC specs. And so you don't have to screen hundreds and thousands of clones in order to get the ones that you need. And then the last piece is the cell engineering challenges. There's lots of ways to edit cells and engineer cells, from viral vectors to CRISPR and other gene editing modalities to mRNA-LNP. And as you saw on that platform slide, we have expertise and we've developed GMP platforms for all of them. And so you don't have to go, to provider X for viral-based approaches. You don't have to go to provider Y for CRISPR and provider Z for mRNA-LNP. We have all the platforms up and running, and we'll figure out which one is the best fit for your program goals. And then that really is summarized by this bottom bottleneck that is another reason, as I mentioned before, that groups like working with us, is we have a pretty comprehensive portfolio of platforms that provide a one-stop shop from project concept to first-in-human clinical data. All right, so another way to look at our iPSC-specific solutions is this collection of hexagons here. So those aspects on the left have to do with the sourcing, reprogramming, and banking of our iPSCs. And on the right have to do with the engineering, basically our CRISPR-focused platforms for engineering the iPSCs. And we'll touch on most of these during the webinar today. So let's start with the front end of this workflow and sourcing the right cells in order to get off on the right start for your iPSC project. And that basically involves a lot of alliance management and a lot of vendor vetting. And so we've done that for you. We have relationships with the key medical clinics that are accredited for iPSC donor material donation. We know the PIs, the staff, and the processes of those medical centers, and we have those relationships in place. We also have built credibility and relationships with the CMS certified CLIA testing labs in order to be able to do any sort of incoming QC as well as product QC before we ship materials to you. And then the last piece of this sourcing puzzle is obviously courier and courier logistics. And we have validated couriers in place to be able to make sure that you get high quality materials that come out of our labs that are still high quality materials when they arrive at your labs or at your clinical centers. So that's the check mark for accessing cells. Let's talk about reprogramming, and we'll spend several slides talking about our unique solution for getting high efficiency reprogramming. So as I mentioned before, our solution is an mRNA-LNP cocktail. The reason that clients like to work with this are highlighted in the bullets here. So first of all, it is a non-integrating approach. So there's no risk, like I said, on that opening bottlenecks to clinical translation slide. There's no risk of genetic integration. And so you don't have that safety risk to worry about. It's incredibly convenient. So we have an optimized collection of 5 reprogramming factors. So it's a slight change from the classic Yamanaka factors. And we've carefully formulated those into a single RNA-LNP cocktail that's ready to use right out of the tube with minimal preparation time before you put it on the cells. It's extremely gentle to the cells, which is always important with any products and processes that you use during iPSC generation. And like I said on one of the opening slides, our clients and our internal team typically see about 4 to 10% iPSC reprogramming efficiency, which really, really cuts down on COGS and on time of screening and characterizing clones coming out of your reprogramming. And it's flexible. We have protocols for using this with feeder-based iPSC generation systems as well as feeder-free conditions. And so a little bit about our mRNA-LNP platform, since obviously the reprogramming cocktail is an mRNA-LNP formulation. So we have a really, really high quality mRNA platform. So in my previous job, I was at Maravai Life Sciences on the strategy and corp dev team there. The best known member company of Maravai is TriLink. So did a lot of work with our both internal as well as external partners around mRNA manufacturing and mRNA CMC challenges. And I can tell you that the platform that uBriGene has made, it really is an industry-leading platform. We get very, very high yields. The downstream team has established some terrific downstream processes for getting rid of double-stranded RNA and getting very homogeneous and pure mRNA. It's not mentioned on this slide, but we also can do circular RNA and self-amplifying RNA for groups who are working in that space. That's not what we have in our mRNA-LNP cocktail. We have classical traditional mRNAs for the 5 reprogramming factors. And the other thing worth mentioning is not only are we able to make, high yields of mRNA in a scalable fashion, but we also offer LNP formulation solutions. And a lot of groups do one or the other well, but our team at uBriGene does both well. And so we have a great platform for doing microfluidic-based mRNA-LNP formulation. We get incredibly good batch consistency. We can program the workflow to give whatever the preferred nanoparticle size is. Usually most of them are around 100 nanometer, but we can regulate that, kind of rheostat that. We get very high packaging efficiencies of the mRNA, almost always in excess of 90%, usually in excess of 95% packaging efficiencies. And it's a very scalable process. We can do this at the milligram scale. We can do this at the multigram scale for mRNA, and it works great in therapeutically relevant cell types like T cells and hematopoietic stem cells, and also iPSCs, obviously. And here's just a little bit about the QC. This is not an exhaustive, our exhaustive QC panel. If any of you folks are into quality or have folks on your quality team that want to learn more about our QC metrics for our mRNA as well as our mRNA-LNP, just let us know, and we can set up a call with our quality team and go over the granular details. But suffice it to say, that we are very rigorous about looking at appearance, pH, identity, purity, potency of each of the factors by themselves. So these represent the mRNA for each of our 5 reprogramming factors. And then once we've formulated them into LNP, we also have very rigorous, and again, this isn't the comprehensive panel, but we have very rigorous mRNA-LNP QC metrics as well to make sure that they meet the specs for identity, purity, potency, and safety that are needed in order to be used by our clients. And obviously for GMP, they're even more rigorously vetted for use in the clinic. So a little bit about the workflow. There's really nothing incredibly unique other than the reprogramming cocktail that we use. We use classic kind of laminin. Our recommended protocol, and the one that we use internally is laminin coated tissue culture dishes. The way that we use our RNA, our mRNA reprogramming cocktail is once the fibroblasts, in this case, this is a fibroblast workflow. We also have PBMC workflows that we can share with you. Once the fibroblasts are thawed and seeded, the very next day, you do daily exposures to our mRNA-based reprogramming cocktail for 8 days. And then after that, you typically change the media. Usually around 2 weeks is when you start to see the emergence of colonies, and I'll show you some photomicrographs of those here in a few slides. And we typically have passage 2 clones ready for cryopreservation within a month. So the workflow is not really changed with our cocktail. It's just that you get a whole lot more positive hits when you're ready to start screening these colonies that you've generated and cryopreserved. Like I said, about a 4 to 10% reprogramming efficiency. So here's some of the photomicrographs. Like I said, so we're treating with our mRNA-LNP reprogramming cocktail daily. In the upper half of these figures, we start to see evidence of iPSC-like cells aggregating around 2 weeks, day 13, and typically by day 18 or so is when you start to see iPSC clones that are ready for picking and initial passage. Obviously, we confirm that we have iPSCs by looking at the classic stem cell markers by flow cytometry, and then we also assess pluripotency of the iPSCs by embryoid body formation assays. So you can see the classic markers for that here, and you can see this data on our website as well. So that's help with the procurement of the starting material, our approach to reprogramming. So let's talk a little bit about our platforms for doing gene editing. So as I mentioned when I was going over the platform slide, we have lots of approaches for getting any sort of payload, including, sorry, I didn't get rid of email alerts, including viral-based approaches, which I won't talk about today, but you can see archived webinars on Lenti and AAV on the website. Obviously, I just gave you a snapshot of our mRNA-LNP platform, and you can get a more exhaustive coverage of that on the website, as well as our archived webinar on that from last September. What I am going to talk about just briefly is our RNP approach. since I haven't gone over that yet. And a lot of people aren't familiar with this. RNP stands for ribonucleoprotein particle. So basically what you do is you pre-complex the Cas gene editing nuclease and the guide RNA into that protein, into that ribonucleoprotein particle, and then you introduce that. We typically do it through electroporation, or we can also, we're working out processes to be able to deliver the ribonucleoprotein particle, that gene editing nuclease guide complex with LNP. We don't have that absolutely dialed in yet, but we're working on that as well. The other thing that is unique about our gene editing platform is the way that we manufacture our guides. So we do not use solid state chemistry to manufacture guides like another company that I worked for previously. But we actually leverage in vitro transcription. So obviously we've gotten really, really good at in vitro transcription. And we talked already a little bit about our mRNA platform. And historically, the way that gene editors generated guides was by IVT, but what they found is that they had a lot of problems with scale, with reproducibility, as well as with cytotoxicity. The cytotoxicity issue is largely because of contamination with double-stranded RNA, which turns on the toll receptors and those sorts of innate immune responses that wreak havoc on the cells. Our team has done an amazing job of downstream processes for getting rid of double-stranded RNA in our IVT workflows. And so our RNA preparations, whether it's mRNA or guide RNA, typically contain 0.01, 0.001% or less double-stranded RNA. So that makes this a very, very simple workflow. It looks a lot like an mRNA workflow because it's leveraging in vitro transcription off of a DNA template. We do digestion to get rid of the DNA template, get rid of any RNA polymerase that's still around. We have purification, downstream purification that yields very high purity guide RNA, and then obviously aseptic filling and cryopreservation. I just want to highlight a few of the other benefits of this approach. I already mentioned that we get very low cytotoxicity due to the low double-stranded RNA. It's a very, very scalable process. So one of the big challenges with solid state chemistry based approaches is, in order to make the amounts that you need for clinical trials, you almost are required, you have to, just because you're limited by the equipment, you have to do parallel runs and then pool those runs. And that is just a nightmare for quality teams, for QC teams, and for the regulators. They don't want to see batches. They want to see a single manufacturing run made of the product that's going to go into the patient. The other limitation that this allows us to overcome is size limitations. So typically with solid state chemistry approaches, about 100 nucleotides or so, you start running into issues with yield when you start doing guides greater than 100 nucleotides. And so, we've done guides 200 nucleotides or longer without any problem. Obviously, IVT is used to make multi-kb length mRNA. And so making guide RNA by IVT gets around that challenge of length limitations for guide RNA. And then the last thing I'll say about this process is it is incredibly green. So one of the things that we're seeing when we talk to clients about this process, they're asking, how much organic waste is involved, yada, yada, yada. Do you have this sustainability metric met? Do you have that sustainability metric met? And the answer to all of those questions is yes, yes, yes. We are, this is a completely organic waste-free process. And so that is attractive to some of our clients who have those types of sustainability requirements in place, not only internally, but in terms of their relationships with external manufacturers. It's become a requirement that they have to meet before they can engage with an external manufacturing partner. And so we're in a great position from that perspective as well. So how do we do RNP? It's fairly simple on a slide, but it's challenging, and it has to be done, every time for every unique gene editing nuclease and every guide with that nuclease, because there is a sweet spot for every guide gene editing nuclease. Cas9 is mostly what we do, but I think you saw on the previous slide we've done Cas12, we've done Cas14, we've done Cas15. We can pretty much do this with any gene editing nuclease that is your preferred enzyme for doing gene editing. But it takes a bit of optimization in order to figure out what the right ratio of guide to gene editing nuclease is. There's also different buffers that are ideal for different enzymes. And then the tangential flow filtration to clean up and concentrate the RNP also has to be optimized depending upon the specific enzyme guide combo that you're using. But we have this optimized and dialed in, and it's very much a platform process. And so what's it look like? The bottom line is we get as good or better gene editing efficiencies as compared to chemically synthesized guides. So the results with our IVT-based guides in the green box here, with classic chemically synthesized guides in the far right-hand column. And these are targets on the left hand that are very common. You probably recognize them. Very common targets for gene editing, usually for improving, hypoimmunogenicity of the cells. The second one is a client-specific target that's confidential that we couldn't disclose. But the top three rows here are iPSCs. The bottom two are T cells. But these are our guide enzyme RNPs delivered with electroporation. And you can see, like I said, we get better or similar gene editing outcomes with our RNP formulations. And the other nice thing about RNP formulations for the folks in the clinic is it just makes it a whole lot easier and introduces 2 or 3 less variables for them to consider when they're trying to figure out what's the right ratio of guide and enzyme to use, etc. If they can get a vial that's ready to go, and all they'd have to do is pop the lid and use it on their patient cells, and everybody's happy with that. And so that's another obvious advantage. So cell sourcing we've covered, reprogramming we've covered, gene editing we've talked about, our ability to do that by virus, do that by RNP, do that by mRNA-LNP. And so the last piece of the puzzle is obviously the seed banking. I didn't put a workflow slide in here, but we can absolutely put an NDA in place, and we can talk a little bit about our workflows for cryopreservation and seed banking. But suffice it to say that we have very rigorous QC like we do for all of our manufacturing platforms. We make sure that the products are safe, potent, pure, and have the right identity and integrity of the active products in them. So this is a pretty thorough QC panel for our iPSC seed banks and master cell banks, and obviously working cell banks we can generate as well. And then so that really leads me to the closing slide to say that we have a very comprehensive toolbox for getting you to where you need to be with your allogeneic cell therapy programs and your iPSC derived cell therapy programs. The one thing that I didn't highlight that we don't do as a platform offering is differentiation. What I will say is we can do it. If you have a dialed in process that you're ready to have tech transferred over, we absolutely are more than open to having our team jump on a call with you and talk about tech transfer and figuring out how to generate, your preferred immune cell type, your MSC or neuronal cell type that you need for your clinical trials. But obviously that's a pretty ad hoc client project specific solution. And so we don't really have a platform per se for offering differentiation services, but we do have the expertise and the capability of doing that if you want us to do that. And so highlights about our iPSC platform capabilities. We pretty much have a dialed in robust process for sourcing and screening of patient materials to get the project started. We talked a fair amount about our mRNA-LNP cocktail, about our platform for generating and doing QC on our mRNA-LNP products. It's a very, very easy to use product with no genomic footprint risks. And that is, like I said, is what we would use if you ask us to engage in a CRO or CDMO type project, whether it be non-GMP or GMP. And then we also offer that as a product that you can use for your own lab's iPSC reprogramming efforts. We have many, many approaches for gene editing that I went over, from virus to CRISPR to mRNA-LNP. And then obviously we have very rigorous QC metrics, the vast majority of which we do internally. I'm pretty sure all of the QC metrics that I showed you for our iPSC banks are internal metrics. Sometimes clients come to us and want additional metrics added on. So, things like teratoma formation or things like NGS characterization of, gene edits that took place. We tend to outsource those types of QC analytics. And then the other thing that we just have announced and launched since the start of the year is we are now offering iPSC banks ready for use for either RUO or GMP applications. And you can find out information on our website about that. And Ming-Juan and I would be happy to talk more about that. And we can get an NDA in place to make sure that we pick the exact right iPSC bank that would be of value to you for your program. And I think that is it. So thanks for your attention. I'll turn it back over to Ming-Juan to lead the Q&A session.
Thank you so much, Scott, for the very insightful presentation. So now we can answer the questions that you submitted. So one question is the reprogramming product GMP manufactured? Basically, if we have offer GMP grade mRNA-LNP reprogramming. Go ahead, Scott.
Yeah, so the answer is yes, and you can see details of that on our website. Looks like Lauren submitted that question. So we can offer our reprogramming cocktail, Lauren, both in a RUO format or in a GMP format. Some groups also, they still prefer to do electroporation as opposed. I'm sorry, need to turn off those alerts. Oh, you're not seeing that now. Never mind. Some people still prefer to do electroporation or transfection-based delivery. And so we also sell our reprogramming mRNAs, so not formulated into LNP. So the answer to the question is yes, we can offer the mRNA in either an RUO or GMP format. And we can also offer the mRNA-LNP cocktail in an RUO or GMP format.
Thank you, Scott. The other question, I think also lots of people are interested, since we have shown the fibroblast reprogramming efficiency is 4 to 10%, which is 10 times higher than the benchmark. So a lot of people are interested in how about the PBMC?
Sure.
What's the efficiency of reprogramming efficiency using PBMC?
Yeah, no, it's a great question, and everybody on this webinar probably knows that getting iPSCs out of PBMCs is more challenging than reprogramming from fibroblasts. So I don't have an exact range. It's usually tenfold or so lower than what we see with fibroblasts. We also have an added product, and it's available on our website as a product for those who are doing reprogramming in their own labs. We call it an enhancer that we use in order to get a higher success rate with PBMCs. But we do have protocols established, and we do have a terrific team that is constantly working on making PBMC iPSC reprogramming easier for our clients and for our internal projects. And so we absolutely could refer you to the website. Looks like Lauren again. And also we can get an NDA in place and talk more granular details about our approach and our strategy for getting high efficiency or higher efficiency PBMC reprogramming.
Thank you, Scott. If you have more questions, you can keep submitting. We have more questions here. Okay, one question is, if they can use the research-grade mRNA-LNP reprogram kit for commercial use, basically if they can create iPSC lines, they can sell it commercially, and also provide iPSC differentiation services as well.
Yeah, no, it's a great question. And the answer is yes. So, we do have licensing associated with groups who want to use, our banks to go into the clinic, and we would have a lot, we would execute a license and have terms and conditions around that for clinical applications. But for groups who are want to use our banks or want to use our reprogramming cocktail for research purposes, even for revenue generating, CRO projects and the generation of differentiated products, we're not going to charge any sort of a reach through or any sort of a royalty or any mucky licensing headaches for you. So as long as you're not making something that's going to go into a patient, we're not going to be chasing you for any sort of royalties or milestone payments or anything like that.
Thank you, Scott. I think that will help a lot of academic researchers, core facilities. Yes, that's a great offer. So one more question is, do you offer lipids for use in LNP formation process development?
Yeah, that's a great question. So obviously that wasn't part of our portfolio, and that's because that's one area that we haven't committed to becoming experts in. But what we have done, the approach we've taken is strategic collaborations. So we haven't really yet made any public announcements about this, but we have a couple of lipid innovators that we collaborate with. And if we get an NDA in place with you and you want to learn, do we have access to lipids that can provide targeting to this tissue or that tissue or have this property or that property, we can work with you and our lipid expert collaborators to be able to figure out what's the right solution for your program.
Thank you, Scott. That's all the questions I have. So if the audience doesn't have any more question, then we are closing up. Thank you, Scott, for answering all those questions. Really appreciate that. As I mentioned earlier, we host a series of advanced therapy webinars on a monthly basis. Please keep an eye on your inbox for information about the next session. Thank you, Scott, again, for a great and informative webinar. And thank you to everyone for attending today's webinar. We really appreciate your time and engagement.
Absolutely.
Yeah.
Ming-Juan, there was one question about cost of licensing, and I'll just say that let's get an NDA in place and we can absolutely discuss that.
Absolutely, yes. So if you have any questions or needs, feel free to email us or call us. And by saying this, have a great rest of your day. And we look forward to seeing you at our future webinars. Thank you so much.
Thanks, everybody.
Bye-bye.